So getting into the next part of the first section of the little bits and pieces that you kind of have to know a little bit about for the rest of the semester to make any sense. And whenever I start a new clump of information, I'm going to put this up so that you can find it. And if you need to get a hold of me, I mean, you can always see me in the lab. You can come in during these times and walk in with a mask and come see me in the lab. I'd call ahead of time. There may be times for one reason or another that I'm not around during these times, but it'll be unusual. I'm recording this at the beginning of August, and I'm pretty much here most of those days. So I won't be here. It's Thursday today. I actually won't be here tomorrow. So there will be days that I may not be around. Anyway, we're moving on. We did the molecules and stuff. Now we're going to get bigger. But we're still not going to get all that big. If this is a protein molecule, then a cell, like our cells, would be Montgomery or Fulton County. Is that there's a humongous jump in relative size between the molecules we've been talking about, even the big ones, and the size of the chambers that the molecules are all cooking in. The picture that I had up last showed I said it was way out of scale, that the molecules wouldn't be jammed together quite the same way. But there's a really, really big difference. And we are now jumping up to the size of cells. Now, some cells are fairly small. One thing that makes bacteria is that their cells are way smaller than our cells. They're simpler in a lot of ways. We'll talk about that when we get to bacteria. And cells have a lot of stuff in them. They have chambers, they have specialized stuff that happens in the chambers. All of that we're not really going to get into. So we're kind of avoiding some of the basic things to talk about. But I do want to talk in general about the bits and pieces. Cells are semi-isolated bags of chemistry. And what isolates them is a lipid-based membrane. We talked about that. We talked about lipids, which has a lot of control over what can go in and what can go out of the cell, and even what can go through parts of the cell, what can go in and out of chambers like the nucleus of the cell. So this is a very, very important thing. I just read an article yesterday talking about the difficulty in getting any kind of drugs that people are developing that they're trying to get into the brain. It turns out that the membranes lining the blood vessels that run through your brain are very tightly connected to one another, so that anything that goes into your brain has literally got to go. In other parts of your body, there's gaps along your tiny blood vessels. Stuff can actually come out through the gaps. But only way into your brain is to go actually through the membrane. So the membrane is much more limiting, what they call the blood-brain barrier. So you may have a drug that will work really, really well on somebody's brain, but you can't easily get it there. Is that you might wind up having to inject it into their cerebral spinal fluid at the base of their spinal canal. That's not a fun way to get any kind of drug delivery, because getting it through the tight membranes of the blood-brain barrier is tough. Even regular drugs, if they got to go into the cell to do stuff, then you got to get them into a cell, and that's not always an easy thing to do. The cells don't always want to let them in. Our cells, bacteria cells don't have this, but our cells has a special chamber for processing DNA and to some extent RNA. Basically, our code centers are in a separate chamber in the cells. That's the nucleus. When we do a microscope lab and we look at our cells, that's mostly what we can see that you can actually point to and go, okay, I know what kind of structure that is. That's a nucleus in a cheek cell. There are other things in there. I'm just going to mention in passing. You have a handout that talks about these in a lot more detail. And so I'll ask you questions about organelles, but I'm not going to talk a lot about them here. And that's the way that the exams work, where some of the questions are coming out of these lectures, coming out of your notes. Some of the questions are coming out of the book, although that won't really be true for a while because the book doesn't really cover this stuff. And a lot of them will be coming out of the handouts, and some of them will be coming out of the lab materials. And you've got a handout that talks a lot about cells and organelles. The little tiny bits of your cells that act almost like organs in a body. They have little special functions that they do for the cell as a whole, the way that your organs have little special. I mean, that's where a name comes from. Somebody went, well, it's kind of like, works kind of like an organ in a body. And went, well, what are we going to call it? And, you know, this apparently was the winner of what they were going to call the little bits and pieces of the cells. A couple of things I want to talk about more specifically. Sometimes the membrane extends out around a structure. And depending on what the inside of that extension is, you've got different types of structures that are involved in various functions that are important to some of the cells. A big extension, bigger than the rest of the cell, I mean longer than the rest of the cell in humans, is a flagellum. It spins like a little propeller. And a lot of you know that this is the movement structure on sperm. There's a lot of other cells in the world. Flagellum is pretty much we just have on sperm. We don't really use them for any other thing in human cells. But we'll talk later about beaver fever with giardiasis. That's a little single cell thing. It's got a bunch of flagella on it. There's a lot of cells in the world that have flagella that allow them to move around. What's kind of a small version of a flagellum, it's built the same way, but they're much, much smaller, and there's always lots and lots and lots and lots of them, are the cilia. There's always so many of them that they actually work very in a coordinated fashion. And again, there's things in the world that swim around using these things. Some of you know what a paramecium is. But in us, cilia primarily line our respiratory system, the tubes, and we produce mucus that lines the tube that catches particles before they get down to the actual breathing part of your breathing system. And cilia is taking that mucus and moving it up to your throat where you swallow it, so that you're always making and getting rid of, otherwise it would cover up with stuff and wouldn't be sticky anymore. So your cilia moves particles out of your breathing tubes, and we'll talk about how that sorts out when we talk about the respiratory system. We have some weird specialized cilia in things like your inner ear and such. We're not going to worry about those. They're modified in a very, very different way. The only other place in a human body where cilia move stuff along is the oviducts, what used to be called the fallopian tubes, where an ovary, when it ovulates, it literally pops a blister in the direction of the open end of the oviduct, and there's a current produced by cilia going down this tiny, tiny, tiny tube that moves the egg cell down to the uterus. And that is the tube where flagella, swimming against the current coming the other way, will find and fertilize the egg cell. And then you have a little embryo bouncing along that is on a little timer, epigenetics, little timer, and will eventually switch on genes and become sticky. And then it'll hit a surface and it'll stick there and start to build a placenta. Why am I going into that much detail? Some of us do expose ourselves to a chemical that paralyzes cilia, keeps them from working as long as the levels are above a certain threshold. It doesn't paralyze them permanently, but if you get enough of nicotine in your system, your cilia exposed to nicotine stop. If you're a smoker, that means that your mucus, catching all the many particles that are in smoke, is not as effective at getting rid of it. More stuff gets down to the parts of your lungs you don't want to get down to. But also, if you raise the nicotine levels in your blood above a certain threshold, the cilia in the oviducts also stop. And so that embryo stalls, and it's much more likely to hit its sticky phase while it's still in that tiny, tiny tube and try to build a placenta there, which it really can't do without exploding the tube. And it can't really build a placenta that's useful. So that is a side effect of chain smoking. People that are always kind of keeping their nicotine levels high enough that the thing stalls. And other kind of smokers do you do find a higher incidence of what are called ectopic, outside the uterine pregnancies in people that expose their cilia to a lot of nicotine. It would be true for nicotine patches as well. Anyway, those are extension structures. Here's a nice Latin word. People saw these amoebas under a microscope, and you pick them up and you move them around and they kind of round up because they're floating and they don't like it. You set them on a surface, and if you watch the cells, a projection will go out from the cell. And on the surface of the membrane, it's kind of tasting what's in that direction. If it likes what's in that direction, it'll stick the projection down and flow into it. And stick another one out. And if it likes that, it'll flow into that. Maybe it decides it doesn't like this direction, it sticks one out in that direction and flows. So they looked at that and went, wow, it's like crawling or walking along, but it's not really, it's kind of making these things on demand. So they're not really feet, but they kind of work like feet. This is Latin for phony feet, pseudopods. Your white blood cells get around your tissues this way. They crawl across the surfaces of the other cells using these projections and stick and projection stick and projection stick. Later we'll talk about this. This is a capability that also turns on in malignancies, in metastatic cancers. This is how cancers spread. Cancers are always kind of randomly turning on different kinds of genes. Some of them are very non-functional and the cells die from it, but gradually in a whole clump of cancer cells, if one or more of them turn on this gene for this ability to crawl, then they will crawl away from the original site of the tumor, and that's when they set up shop other places. Now you've got a spreading cancer, that's not good. But they never turn on flagella to do it, they never turn on cilia to do it, they only turn on pseudopods to do it. One would assume because it's a simpler genetic switch, and that capability turns on. We'll talk more about cancer later. There's one other type of extension structure I want to talk about. Microvilli literally means little hair in Latin, little hairs. These look like cilia. They coat the linings of places where you need to move stuff through the surfaces. Is that a whole bunch of these on a surface makes for a lot more surface than just the flat edge of a cell. So it increases the surface area. Your intestines are covered with this stuff. Tubes in your kidneys that are pulling stuff out of your blood and putting it into urine or putting stuff back in that came out through the filters that you want to return are covered with microvilli. It's a way of taking a limited surface and really increasing the surface by extending and extending and extending and extending it. And they don't move the way the cilia do, they basically just wiggle in order to help get stuff down in between, but they're not moving things like cilia do. Okay, from here we're gonna move to tissues. So in my hope of trying to keep these fairly short, I'm gonna stop here and pick the tissues up next.